All right, it is 2 o'clock, so we will get started. Just a reminder for where we left off on Wednesday, we talked about the energetics of folding when it comes to a folding funnel. And then we talked about how disulfide bonds can be incorrectly formed when a protein is trying to fold. And you need enzymes called protein disulfide isomerase to come in and change the disulfide bonds to their native state. Just while I'm looking at it right now, isomerase, anytime you come across an enzyme that's called an isomerase, the name is telling you what it's doing. It's just switching bond orders. That's basically what it's doing. It's switching how things are bonded to each other. So a protein disulfide isomerase, by the name, is taking a disulfide bond and just switching how the molecules are bonded to each other, right? So that's where we left off on Wednesday. Here, to begin with, on Friday, we are going to talk about chaperones. So a chaperone is a protein that helps to refold proteins that have not folded properly. So they will find misfolded proteins and try to help enfold them. The reason why we need chaperones is that if a cell has a lot of unfolded proteins, they can start to aggregate together. So remember when we talked about on Wednesday, the first thing to fold, or one of the first things to fold in a protein, is all the hydrophobic amino acids will go towards the center of the protein. This is due to the hydrophobic effect. Well, if a protein doesn't fold correctly, then you will have hydrophobic amino acids that are just showing to the water, right? They're not hidden away within the protein. And water, as we learned, doesn't want to interact with hydrophobic side chains because they can't hydrogen bond to them. So they're going to try and push them together to other hydrophobic residues and form that clathrin. And the unfolded proteins, they'll start pushing all these unfolded proteins together until you get like a big ball of unfolded protein that's just in your cell. And this can lead to cell death when you have this huge unfolded protein mass. So to prevent that, to prevent unfolded proteins from aggregating with each other and to prevent cell death, we have these chaperones that will go and bind our unfolded proteins and try to fold them. The protein we are looking at today, our chaperone, and it's not the only chaperone, but it's one of the well-studied one. It's called the GroEL, GroES system, right? So this interacts with proteins that are anywhere from 20 to 60 kilodaltons. Usually looks at alpha beta domains, and we'll talk about those today. Looks at folding proteins with beta sheets. Anything that's like a typically slower folding protein, GroEL, GroES can help fold it. So I'm going to start in the top left to explain this picture. And what we're looking at here is when we talk about GroEL and GroES, they are actually two separate proteins. GroES is like a little hat, and GroEL is the chamber, right? And when inside the cell, two separate GroEL molecules will come together to form one unit. However, there is a barrier between the units. So an unfolded protein can only can only bind to one GroEL at a time. You can never have a protein in each chamber at the same time. Only one protein will enter at a time. All right, so we're starting on the top left. Unfolded or misfolded protein will go and bind inside of GroEL. And GroEL will have ATP associated with it. After you have bonded or captured the misfolded protein, the GroES hat will come and sit on top of the GroEL chamber. This will help and trap the protein within GroEL. And this also causes a conformational change. So the shape of GroEL actually changes when GroES binds to it. It goes to what we call the trans ring form to the cis ring form. In the cis ring form, we can start the fold now. So this conformational change will also lead to the ATP that is bound to GroEL. All of these will hydrolyze. They'll lose a phosphate and become ADP. And it's with that hydrolyzation event that the protein starts to fold. And so now we have a nice folded protein. After this hydrolyzation event, a second separate misfolded protein will go and bind in the second GroEL chamber. And 7 more ATPs will bind to this second GroEL chamber. Once the second misfolded protein is bound, GroES will come off, the ADP will come off, and your protein that is now folded correctly can leave, and this process starts again, except here, the image becomes flipped. So this red, just imagine in your head, it's flipped 180 degrees, and then we start over again. So that's basically how the GroES, GroEL system works, right? Bind a misfolded protein, put on your GroES cap, change into the cis ring, hydrolyze your ATP. Have the protein folded inside the cis ring, bind a new misfolded protein in the other GroEL, take off your GroES cap, remove the ADP, remove the better folded protein, start the process again with the other misfolded protein. So before I get to these questions on the side, anyone have any questions on how the GroEL, GroES system works? All right, so let's go to this question. So here's kind of a critical thinking, big picture question. So before GroES attaches, so before this hat comes on, we want to capture a misfolded protein. Therefore, what type of amino acids are in the GroEL chamber before the cap comes on? And what type of amino acids are in the chamber after GroES attaches? Does anyone have any ideas what type of amino acids would be inside the chamber before GroES attaches, and what amino acids are there after GroES attaches? Good old critical thinking. Well, maybe to help you along, let's remember what type of amino acids we have, right? We have charged amino acids, so your positive and negative charge. We have your polar amino acids, and we have your nonpolar amino acids. All right, so those are the three types of amino acids we have. All right, and if you're an unfolded protein, who can remind me, if you're unfolded, what amino acids are you showing to the environment that you should normally have hidden away if you're folded? Are those charged amino acids, polar or nonpolar? Hydrophobic, right? So if I'm trying to capture this misfolded protein in GroEL, what would associate with these nonpolar amino acids? Should I have charged amino acids in there? Should I have polar amino acids in there? Or should I have nonpolar amino acids in there? Right, nonpolar. So that's the idea here, right? To capture a misfolded protein, you would want to have hydrophobic amino acids in GroEL, because the hydrophobic effect is water will just naturally push another misfolded protein to you. That's how you capture something. Then when I want to fold a protein, I put in my GroES hat and I want to simulate the aqueous environment of the cell. So when I burn those ATP, what in fact GroEL is doing is that it's rehiding all the hydrophobic amino acids and now showing the misfolded protein polar amino acids to mimic the water environment of the cell. And what GroEL is trying to do when doing that is it's trying to get the hydrophobic amino acids to go back into the core and to have the polar charged amino acids on the outside to have a folded globular protein. So any questions about that logic or how GroEL, GroES works? All right, and so in B, we have this question. Why do some heat shock, so heat shock proteins, heat shock protein is similar to the idea of a chaperone, except it's a much wider class of proteins in that there are heat shock proteins that are chaperones, but not all heat shock proteins are chaperones. So these are just proteins that are turned on under stress conditions, so like heat. So when a cell starts to heat up, it'll start making these proteins. So why do some heat shock proteins speed up degradation rather than refolding? So I'll answer this one, right? As you increase the amount of heat inside of a cell, you are increasing kinetic energy, and proteins are barely stable as is. So as you increase heat, you increase the number of unfolded proteins, unfolded or misfolded proteins. And as I stated when I was talking about GroEL, GroES, if you have a lot of unfolded proteins, they can aggregate together and cause cell death. So to prevent cell death, what some heat shock proteins do is that they will cut up the unfolded proteins. You know, let's just think of like taking two knives and just going. That prevents this big ball of aggregation, which prevents cell death. So it's kind of a last ditch effort to keep the cell alive. Just chop up any misfolded proteins. I don't have time to try and refold them. It's a critical moment in my life. Destroy them so I could live, hopefully. So any questions about that idea with heat shock proteins or anything presented here? All right. So let's talk about some protein folding diseases. And we know roughly at least 35 human diseases that are due to misfolded proteins that are aggregating. And these are called amyloids, and the disease are known as amyloidosis. What we're seeing down in the bottom left here is an example of amyloids. They're basically beta sheets that stack up on each other, and they stack up more and more and more to cause these plaques. And these plaques, when they're around healthy cell, will kill healthy cells. And one of the most famous amyloid diseases is Alzheimer's, caused by amyloid beta. Now, most everybody in the world has amyloid beta. They make this protein. However, we don't know what it does yet. We just know that it's there, it's present, but we don't know its function. We don't know what it's doing. What can happen in some people is that this amyloid beta can start to form plaques in the brain tissue. Like I said, when you have these plaques, it'll kill cells, and so it can kill your brain neurons. And each of these plaques, 40 to 42 residues of beta sheets. And what's really bad about Alzheimer's is that, if you are prone to get Alzheimer's, you are most likely making those plaques right now as we speak. The deposition is an ongoing process, most likely, that as you age, you get more and more and more of these plaques. And that's why we see Alzheimer's affecting elderly people, is because they just had years and years and years of these plaques growing, killing more and more neurons until you start misremembering things and have a hard time with memories and recognizing other things. And really, the treatment for Alzheimer's right now is, can we find a way to stop making this protein? Because the idea is, if you don't make any amyloid beta, right, you're not going to form these plaques. If you're not going to form these plaques, then you don't have Alzheimer's. So is there some way that we can slow down or even stop this protein? And it should take care of that disease. So any questions about prion diseases, or anything up to this point? All right. And actually, these prion diseases, we have found that there are transmissible versions of them. The most famous being mad cow disease. If you remember back, they had to kill a ton of cows, millions and millions of cows in England, because these cows had a transmissible spongiform, I can't even pronounce that word today. Sorry, my brain is not there. TSEs. They're basically prion rods that you can transfer between organisms. So, you eat meat from a cow that had prion rods in them. You digest these prion rods. They go into your body, and they serve as a basis for amyloid plaques to form. So that's why it can be dangerous for us. Another type of disease from transmissible prion rods is called kuru, or kuru. It's a brain disease from cannibalism. Again, it's the same idea. You eat somebody who has prion rods, and then you get them yourself. And these prion rods, hydrophobic, very hydrophobic. That's why they serve as a starting point for a plaque, because other hydrophobic amino acids or proteins will start to combine with them. And the aggregates are called amyloids, like we talked about in the last slide. And the scary part is we create prions as well. Again, we don't know what they're doing. We just know cells make them. And if you have a prion called the scrapie prion or PRP, you can cause an aggregation of these prions. So PRP kind of serves as the as like the starting place for creating prions, which means if you don't have PRP, you are immune to all these prion diseases, which is great. But, you know, it's kind of a crapshoot if you don't have this or not. So those are prion rods. And those are the different types of prion diseases, proteins caused by diseases that you can get. And that is it for this slideshow. So we're going to go to the slideshow that I made for Friday. And I do apologize. I just realized today when I was going over stuff, I kind of mixed up the order. Like the PowerPoint we were going over on Wednesday should have been the PowerPoint for today. But it will still work out. So we were talking about protein folding. And so that is tertiary structure. Like I said, it is the overall 3D structure of proteins. And how do we actually know what these proteins look like? We talked about the folding funnel, and we talked about these higher prion diseases that can happen, but how do scientists know, what does the 3D structure of a protein look like? We can't see them with the eye. And so one way to determine the 3D structure of a protein is to shoot it with a very high-powered laser. That's called X-ray crystallography. And the idea of X-ray crystallography is that you take a protein and you make it extremely highly concentrated. So you get a lot of this protein in a very small volume. And then you try hundreds of different conditions. And I'm talking about like different salts, different solvents, different temperatures. You just try a bunch of things with this highly concentrated protein solution you have, and you're trying to get a crystal. And it's not easy. And there's a lot of times no rhyme or reason why something works. It's just sometimes you get a protein crystal after your 1,000th try. But the idea here is that we're trying to see the structure of this protein crystal. So what you do is you take this crystal and you shoot it with an X-ray while moving it 360 degrees. This is the high-level version of this is done in a synchrotron. So if you're not sure what a synchrotron is, I'll draw a crappy little picture. So what a synchrotron is, it's a giant circle. And around these circles are magnets. And what you do is that you shoot electrons around this circle, and then you have an atom, like just sitting in the pathway. And you bombard this atom with very high-powered electrons. The idea is you're trying to rip this atom apart to see what it's made of, trying to understand dark matter and all those high-level physics experiments. So that's what a synchrotron is. The cool thing about a synchrotron that biochemists can use is that when your electrons are going around in a circle on 90-degree axis from the circle, you shoot off X-rays. These electrons moving fast in a circle naturally just shoot off X-rays. So at a synchrotron, what you'll see around like the giant circle are little buildings. These buildings are for biochemists and chemists to harness the X-rays that are coming off of these synchrotrons for X-ray crystallography. So you have this crystal spinning at 360 degrees slowly. It's very slow how you spin it. and you hit it with X-rays. You obtain what's called a diffraction pattern. So what happens when you hit a protein crystal with X-rays is that if the X-ray hits a nucleus, it will bounce. And we capture how the X-ray is bouncing on film. So this is an actual result from an X-ray crystallography experiment. This giant hole in the center, that's the X-ray beam. All of these spots are X-rays being bounced when they hit an atom. And you can do math and determine based on this picture, where are the atoms in 3D space? The math isn't actually that hard. It just takes, a couple weeks to get used to, and then you can get it. We're not going to look at that math. That's physical biochemistry. It's cool, but we're not going to look into it. What we can do though is we can feed this into a computer and do what's called a Fourier transformation. Fourier transformation. And these dots become a 3D image. So you get this 3D electron density. Let me get a different colored pen so I can write on that black. Sure. So these little cages are their electron density. And that's just telling you, where are atoms? And so after you get this electron density map, you try as best as you can to fit your primary structure, remember, is the sequence of amino acids from N-terminus to C-terminus, into this picture. And it's kind of like a video game at that point. You are trying to get the highest possible score you can. And the way you get points is by making favorable interactions based on the rules of biophysics. You're making sure that your angles are good, your dihedrals are good, you have favorable positive, negative interactions. Anyone who is interested in looking something at doing something like this, and what I would say is go look at Folded at Home. I believe that's what it's called. Folded at Home is a way for novice scientists to work with protein structures and protein folding. It's out of Washington. And what they're trying to do is they're trying to use the manpower of the internet to solve really complicated biochemistry problems, like how does this protein folded? And they've actually released some papers with like the highest scoring players as first authors. Like they released a paper with like 3,000 people on it, and it's all the users who helped to fold that protein. So really cool software, really cool idea there. So for X-ray crystallography though, the only way you get these spots is if your atom is not moving in your crystal. So that's why this question, X-rays usually cannot reveal the first few or the last few residues of a peptide. Why? That's because in a folded protein, generally what happens, if this is my folded protein, the N-terminus and C-terminus are not folded. C-term. They're usually like floppy wet spaghetti, and they're just moving all over the place. So since you have 1,000 copies of a protein, let's say in that crystal, and every single protein has the N-term and C-terminus in a different position, we don't get enough of a signal to see a spot on this map. The only way we see a signal is through amplification, where if we're looking at 1,000 proteins, like an atom is always in the same location in that protein. It's not moving. That's how we get the signal amplification. That's how we can get this 3D map. But if there's a lot of movement, like in the N and C termini, you can't actually get any diffraction. You can't get this 3D map. All right, so that was quite a bit of information on X-ray crystallography there. Does anyone have any questions about the process? All right, let's move on to our next technique to get structure. You might be asking yourself, why would people bother with getting structures of proteins? And the phrase I want you to keep in mind is that structure determines function. To really know how a protein works, you have to know what it looks like. And then once you have the structure for a protein, you can start designing drugs against it if it's a problem, like the COVID virus. To really try and inhibit, one way to stop it is to inhibit it. And the only way to inhibit it is if you know what it looks like, because then you can start targeting like specific proteins. So that's actually how we fight some viruses, is that we just inhibit the proteins in it. Anyways, let's talk about our next way to determine protein structure. Is everyone's favorite from organic chemistry, NMR. And if you thought that the spectra you get in organic chemistry were bad, let me show you a NMR from biochemistry. So try and look at that and get structure, which is what we do. And in grad school, one of the first problems in the first class I took was to get something like this and just do it by hand and tell me what is the 3D location of stuff. So that was fun. So NMR. What NMR is, it's nuclear magnetic radiation. And this technique works because certain nuclei absorb radio radiation if they're in a magnetic field. And the way they absorb this radiation will change based on its local surroundings. So by doing different experiments, you can tell how other atoms are in relation to each other in 3D space. You can tell what's around other things. Couple different experiments that are commonly done in biochemistry is called the COSY, or correlation spectroscopy. What a COSY is that you look at how far hydrogens are away if they're connected by one or two atoms. So in a COSY, I'm measuring this distance, right? And so if I do a COSY type of experiment for like every single pair of hydrogens, I can actually start to map out, okay, where is every single hydrogen in the protein? Because if you can map out where every single hydrogen is, you can map out the rest of the protein as well. And you also have what's called a nosy, and this is the distance for proteins that are close in space, but it's interatomic. What that means is that they're not bonded. So actually, I do something like this. So if I have a carbon and another carbon in my protein, but they're not connected, but they're close, I can tell what the distance is. So what you do is that you do a lot of these measurements and you get all these parameters. And then you try and make a protein that fits these parameters, rather a 3D structure that fits these parameters. And you also have other chemical constraints such as bond distances, angles, if something's in a plane or not, chirality, van der Waals radius. You feed all of these parameters into a computer. And then you get what's called as an ensemble. So I know this question is kind of cut off here. But when you do NMR spectroscopy, you will get at least 10 structures back. That's what an ensemble is. However, if you compare this with X-ray, you only get one structure back. based on how I described these techniques, NMR versus X-ray, can anyone determine or think why when we do X-ray, we would only get one structure back, but when we do NMR, we can get like 10, 20 back? Does anyone have any ideas about that? Why in NMR do you get multiple structures, but in X-ray you only get one? Well, let's think about how NMR is done. So hopefully everybody's studying NMR in organic chemistry. If you took it through Tamusa, I know Shelton has an assignment where you have to figure out a spectra. So hopefully you talked about how it was done. So what media is NMR rather done in? Do you use like gas? Do you use liquid, or do you use solid to do NMR? Or if somebody's done NMR before, you should know this as well, if anyone's had the chance to use one of those machines. Done in a gas, a solid, or a liquid? Anyone have an idea? If you don't want to share publicly, you can always send me a private message. Well, in case you didn't learn that in organic chemistry, or you forgot organic chemistry, NMRs are always done in a liquid, which means that when you do NMR, you have proteins that are moving. They're doing what's called breathing. Their proteins are flexible, so they'll breathe like in and out almost. So that's why you get multiple ensemble structures in NMR, is that you take multiple snapshots of slightly different proteins. While in NMR, you're in a crystal, so you're more or less frozen. So you're frozen in a crystal, you can't really move. In NMR, you're in a liquid, so you have a lot more movement. So that's why you get a lot more different structures. All right. Next type of structure determination experiment that you can run is called Cryo-EM, called cryo-electron microscopy. And what you do is you take a sample that's dissolved in water, so a protein sample, and you just freeze it extremely fast to minus roughly 200 C. When you freeze a protein water solution that fast, water doesn't have time to form ice. You lower the temperature so fast that it just kind of stops, like moving, but doesn't rearrange to form ice. And that's why I'm saying it's like this glass-like state, which holds the protein in place, but doesn't destroy it. When a protein is frozen in water, a lot of the times the protein will be destroyed. But if you just super freeze the water, that the water doesn't turn into ice, then the protein will just stay there in the water. And it will be in its native state, and then you use electrons to just try and get an image of the molecule. This won the Nobel Prize 4 years ago in chemistry. And here on the right is an actual structure from Cryo-EM, apoferritin. On the bottom here, these little squares, that's GroEL that we talked about previously today. So that's just another way that you can get structure determination through Cryo-EM. All right. So from all this structure and what we talked about on Wednesday, we have some general ideas of what happens to different amino acids when a protein folds. So any nonpolar residue is going to be in the interior of the protein, called the hydrophobic effect again. Water doesn't want to interact with hydrophobic amino acids, so it's just going to shove them in the center of the protein. Anything that's charged, positive or negative, that's going to be on the outside, because charged molecules want to be around other molecules in opposite charge. So that's why they're just going to be surrounded by these molecules. It's very energetically unfavorable for them to be buried. And then your uncharged polar groups, they're usually on the exterior, but they don't have to be. If they're on the inside of a protein, they have to be hydrogen bonding with something. Again, because the inside is usually hydrophobic. So just to make sure we understand what a hydrophobic amino acid is, I have a question here, which I think I have to pull for. Perfecto. Yeah. So I have 4 peptides here. Which peptide do you think is most likely to be on the inside of a folded protein? So who can remember their amino acids, amino acid properties? All right. So looks like we have the majority who has it. Yeah. So the hydrophobic, I almost said aliphatic. The hydrophobic amino acids would be number 2, val, isoleucine, leucine, tryptophan, alanine. Asp, charged, asparagine, glutamine, polar, tyrosine, a low polar, not really though. Lysine, charged, serine, polar. So number 2 would definitely be in the center. And so B, based on what we just said, proteins are usually in water. What would happen if you took a folded protein and then put it in like oil? Because they want to have an idea what would happen there. Now, guys, along those lines, is there a hydrophobic effect in a nonpolar solvent? There's not. So what would happen to the protein? Would the protein remain folded? Or would it go unfolded? Because remember, the whole thing that drives protein folding that we kind of talked about on Wednesday and just right now is the hydrophobic effect. That is what is pulling, that is what is pushing all your hydrophobic amino acids towards the center of the protein. So yes, if you took water away from the protein and put it in a nonpolar solvent, like the reverse would happen. All of the charged amino acids would no longer have anything to interact with. So they would go and find each other and interact within the protein. And so the hydrophobic amino acids in the center, they have no reason to stay in the center. It was water driving that process. So they would start to unfold. So if you put a protein in a nonpolar solvent, it would unfold because you have lost the hydrophobic effect. The hydrophobic effect is what drives protein folding by shoving these nonpolar residues in the center. And then our last question, I guess I'll go ahead and answer this. So if we have a membrane, a lipid bilayer, some proteins, and I'll draw one here, some proteins go all the way through the membrane. How would the position of amino acids be different in these proteins? Here, the hydrophobic would have to be on the outside, right? Because inside the membrane, your lipid tails are very hydrophobic. So anything that passes through this membrane also has to be hydrophobic to have your hydrophobic interaction. And you need polar amino acids at the head groups here. But that's how it would mainly be different for any protein that travels through the membrane. So any questions about information presented here or anything else we went over today? All right, so it's 2:48. I don't think I have enough time to go on to another topic within, because I can't cover something in 2 minutes. So we'll call it here. If you do have questions about anything, please don't hesitate to email me. I will put up a homework up as always. Other than that, I hope you have a good weekend. Thank you for stopping by. I always appreciate it. So I'm not talking to myself in an empty room by myself. And I will see everybody later. Have a good weekend, everybody.